I was looking through some recent medical data when I stumbled upon a statistical anomaly that feels almost like a dark joke from nature. For years, doctors have noticed a strange, recurring pattern. People battling aggressive, terminal cancers, the kind of diagnoses that feel like an immediate death sentence, hardly ever seem to develop Alzheimer's. It's a bizarre, unintentional trade-off. It's as if having one of the world's most feared killers circulating in your body somehow shields your brain from the slow, agonizing fade of dementia. The prevailing medical thought has always been that these are two separate wars, two distinct ways the body breaks down as we age. We view them as the two great villains of modern medicine. But a team of researchers in Wuhan recently decided to look closer at the plasma of these cancer patients, and what they found defies our traditional good versus evil logic. It turns out those deadly tumor cells, the ones we spend billions of dollars trying to eradicate, are secreting a specific protein called Cystatin C. Now here's where the narrative takes a sharp turn. For the last 40 years, the entire pharmaceutical industry has been obsessed with a single strategy, trying to shut down the factory. They focus almost exclusively on preventing amyloid plaques from forming in the brain. But the results have been underwhelming, leaving families with little more than a slightly slower decline. This cancer-derived protein, however, doesn't care about the factory. It acts like a ruthless, specialized cleaning crew. It doesn't just stop new plaques from forming, it hunts down and dissolves the garbage that's already there, effectively scrubbing the brain clean. It's a radical, modern twist on the ancient concept of fighting poison with poison. We've spent decades viewing cancer as the ultimate biological enemy, but this discovery suggests a disturbing yet hopeful irony. What if the very thing trying to kill the body is carrying the molecular key to saving the mind? It forces us to ask a question that felt impossible until now. Could the cure for our longest goodbye be hidden inside our most violent disease?
It's a question that really just shakes the foundation of how we think about disease, doesn't it?
It does. Because you hear that, and it sounds counterintuitive. Uncontrolled cell growth in one disease, protecting against uncontrolled cell death in another.
But the data is there.
Yeah.
And it's not a small effect. When you look at these huge population studies, I mean, we're talking tens, even hundreds of thousands of people followed over decades, the pattern just holds.
It's consistent.
If you have a history of cancer, your risk of developing Alzheimer's is significantly lower. And maybe even more bizarrely, if you look at it the other way around.
Alzheimer's patients don't get cancer.
They have a surprisingly low incidence, yes.
Yeah.
It's like a biological seesaw. One side goes up, the other has to go down. It's a trade-off.
Okay, but I have to play devil's advocate right away.
Of course.
Isn't this just a grim statistical illusion? Survivorship bias. If you get a terminal cancer diagnosis at 65, you don't live long enough to get Alzheimer's at 80.
That is the first and most important objection to raise. And every serious researcher in this field has had to grapple with it.
Because it seems like the most obvious explanation.
It does. But the good studies, the really robust ones, have controlled for age. They're not just looking at raw numbers. They're comparing, say, an 80-year-old cancer survivor to an 80-year-old who has never had cancer. Even then, the protective effect remains. The cancer survivor is still less likely to develop dementia, so it's not just about dying early. There's something about the underlying biology, something about the cancer itself, or maybe the body's fight against it, that is changing their risk profile for neurodegeneration.
So the body it's almost like it has to pick a mode. It's either in grow and survive at all costs mode, which leads to cancer.
Or it's in prune and delete mode, getting rid of dysfunctional cells, which, if it goes too far, leads to neurodegeneration.
You can't be great at both.
Nature struggles to optimize for both, especially as we age. It seems you get a body that's really good at stamping out rogue cells before they become tumors, or you get a body that's really good at cellular repair and regeneration, but that same vigor can become cancerous.
Okay, so sure, let's get into the mechanism, the how, before we tackle the deeper why. I mentioned that protein from the opening, Cystatin C.
Right, CCS.
It sounds like just another lab chemical, but this is something our bodies are making. So what is it actually doing? How does a protein made by a tumor in the lung, for instance, protect the brain?
To really get that, we have to talk about the villain first, amyloid plaque. But even that is a bit of a misnomer.
So?
Well, the plaque is the tombstone. It's the big, ugly, insoluble thing we see on an autopsy. The real damage, the thing that actually kills the synapses, happens much earlier.
Okay.
It starts with these little proteins, these peptides called amyloid beta. They float around as single units, monomers, harmless enough. But then they start to clump together, first into these small, soluble gangs called oligomers.
And these are the bad guys.
These are the bad guys. The oligomers are incredibly toxic. They interfere with synaptic function. They trigger inflammation. They're the real killers. Then eventually those oligomers stack up until they form long, hard fibrils, and those fibrils become the plaque.
And for 40 years, all the drugs have been aimed at either stopping the production of amyloid altogether or, trying to break up those big plaques at the end.
Right, trying to clean up the tombstones after the damage is done. And it has been, as you said, a story of underwhelming results.
So where does Cystatin C come in?
Cystatin C is what's called a protease inhibitor. In simple terms, it's a regulator. It tells other enzymes, hey, stop what you're doing. Specifically, it inhibits a family of enzymes called cathepsins.
Cathepsins?
Think of them as part of the cell's garbage disposal system, the lysosome. They break down old proteins. But in an Alzheimer's brain, that whole system gets out of whack. It's not just failing to clean, it's actually contributing to the toxicity.
Okay, so it regulates the cleanup crew.
It does, but its most direct role here, the really elegant part, is that Cystatin C can bind directly to the amyloid beta peptide itself.
So it grabs it?
It grabs it. Think of it like a chaperone at a school dance. It sees a few troublemakers starting to gather in a corner, and it steps in and separates them. It says, you are not allowed to clump together. It physically prevents that toxic oligomerization step.
It stops the gang from forming before they can do any damage. It's not cleaning up the mess, it's preventing the mess.
The evidence is really strong. You take those mouse models of Alzheimer's.
The HAPAP mice?
HAP mice, right. They're genetically engineered to overproduce human amyloid. If you then go in and you genetically knock out their ability to make Cystatin C, their brains just fall apart.
Really?
The plaque load explodes. They develop severe cognitive deficits way earlier than their litter mates who can still make the protein. It's like taking the brakes off the system.
Because they've lost that chaperone.
They've lost the check. Let's go back to the cancer patients. Tumor cells are metabolic furnaces. They are hyperactive, just pumping out a storm of proteins to help them grow, invade tissue, and hide from the immune system.
All for their own survival?
For their own selfish, malignant survival. And one of the proteins they happen to churn out in huge quantities is Cystatin C.
So the tumor is flooding the person's entire system with this protective chaperone protein.
That's the hypothesis. The tumor using Cystatin C for its own reasons, maybe to break down tissue barriers so it can spread, but a side effect of that is that the blood becomes saturated with this stuff.
And it gets into the brain.
The blood-brain barrier is not a perfect wall, especially as we get older, it gets a little leaky. So this systemic flood of Cystatin C provides this collateral benefit. It creates an environment in the brain where it's just very, very difficult for amyloid to start clumping up.
It's an accidental shield.
An accidental shield. And this isn't just in mice or cancer patients, it's in our genes. There's a gene called CST3 that codes for Cystatin C. And there's a common variation, a polymorphism, that leads to your body just secreting less of it.
Yes. People with that variation
have a significantly higher risk for late-onset Alzheimer's. Less of the chaperone protein means more risk.
It's incredible. We have the mechanism, but that just brings up the bigger, almost philosophical question. Why would our bodies be built with this fundamental trade-off between cancer and dementia? This is where evolution comes in.
Mechanisms like this don't just appear out of nowhere. They are the result of millions of years of selective pressures. And it forces you to confront a really uncomfortable truth. Our bodies were never designed to last forever. They were designed to perform.
To perform within a certain window.
Exactly. And that's where we get into these deeper evolutionary theories.
I want to frame this with the story I found in the research, a kind of case study. It's about a man named Elias.
Yes. Elias is the perfect illustration of the first major theory here, which is called antagonistic pleiotropy.
Mouthful.
It sounds complicated, but the idea is actually very simple and very powerful.
So let's talk about Elias. He's a construction foreman in Chicago in the 1950s and 60s. This is peak, high-rise boom, dangerous work, no OSHA, no safety harnesses. It was pure physical grit.
A high-risk environment.
Absolutely. And by all accounts, in his 20s and 30s, Elias was a physiological Superman. He was the guy who could smash his hand with a hammer and the swelling would be gone in 2 days. He'd get these nasty cuts that would just seal themselves up. He even broke a bone and was back on the scaffolding in a few weeks.
So his body was just exceptional at repair, a very high-performance system.
Exactly. If you could have run his blood work back then, you'd probably see sky-high levels of growth factors, a really aggressive immune response, rapid cell turnover. From an evolutionary standpoint, he was a winner. He was strong, he was resilient, he survived to have 4 kids by the time he was 28.
He was absolutely winning the game. Natural selection would have strongly favored whatever genes were giving him that edge.
But the other side of the coin appears. He retires at 65, he's healthy, looking forward to his golden years, and within 2 years, the decline was just catastrophic. It wasn't a gentle slide into forgetfulness, it was a rapid, aggressive, full-blown dementia. The man whose body could heal itself in a flash couldn't figure out how to tie his own shoes.
A complete betrayal by his own biology.
And how could a body that was so robust, so good at survival, suddenly become so fragile in the mind?
And that is the core of antagonistic pleiotropy. The term was coined by George Williams way back in 1957. Pleiotropy just means a single gene can influence multiple seemingly unrelated traits.
Okay, one gene, many jobs.
Right. And antagonistic means those jobs work against each other over time. So you have a single gene that gives you a benefit early in life, but causes a problem late in life.
So the same gene is both good and bad.
It's good when it matters for evolution, and bad when it doesn't. Think about a gene that promotes rapid cell division and repair. For Elias, in his 20s, that gene is a lifesaver. It heals his wounds, fights infections, keeps him strong. It ensures he survives long enough to pass on his gene.
Evolution loves that gene.
It selects for advantage. But what happens when you have a system that's built for rapid, aggressive cell division and you let it run for 70 years?
I am guessing that's the definition of cancer risk, isn't it?
Or let's flip it. Consider a gene like p53. It's called the guardian of the genome, and it's a tumor suppressor.
Yeah.
When a cell gets damaged DNA, p53 slams on the brakes. It says, stop it dividing. Either repair this damage or you die.
So p53 prevents cancer. It's a good guy.
Essential. But what if you have a version of p53 that's, let's say, overzealous? It's so good at stopping cancer that it's trigger happy. It tells cells to die, to undergo apoptosis, even for minor damage. Over the first few decades of life, that's great. You have very low cancer risk. But over 70 or 80 years, that constant aggressive culling of slightly damaged cells depletes your stem cell reserves. It leads to tissue breakdown, to atrophy. It means your brain loses its ability to repair and replace neurons.
So the very gene that protected him from cancer in his 30s is what's driving the neurodegeneration in his 70s.
That is the antagonistic trade-off. And here's the cold, hard part. Evolution doesn't care about his dementia.
Because he already had his kids?
He already had his kids. The negative effect of the gene shows up long after he's passed it on. Natural selection is effectively blind to diseases of old age. As long as the gene got you through your reproductive years, its job is done. The biological warranty expired.
It reminds me of those studies on Huntington's disease.
A perfect, though tragic example. Huntington's is a devastating neurodegenerative disease caused by a single gene.
A death sentence.
But when you look at the data, people who carry the Huntington's gene before they show symptoms actually have lower rates of cancer and higher fertility rates.
Ow.
The gene seems to ramp up that p53 activity. So it gives you a reproductive and survival advantage in your 20s, only to exact this terrible price in your 40s or 50s. It is a Faustian bargain written directly into our genetic code. I will give you vigor now for your mind later.
And evolution takes that deal every single time.
Without hesitation.
Which brings us to the second big theory, which feels even colder somehow. It's less about genetics and more about, well, accounting, the economics of staying alive. And for this, I want to talk about Sarah.
She represents the other side of this coin, the disposable soma theory.
So Sarah is 95 years old. She lives in a state-of-the-art, climate-controlled assisted living facility in London. She's had access to the best of modern medicine her whole life. Vaccines, antibiotics, surgeries, excellent nutrition. Physically, for 95, she's okay. Her heart works, her lungs work.
Modern medicine has kept the machine running.
The machine, yes, but the operator's gone. She hasn't recognized her own daughter for 5 years. She is neurologically absent.
And the disposable soma theory explains why this is not a surprise, but an expectation.
It feels brutal.
It's pure thermodynamics.
Yep.
The theory was developed by Thomas Kirkwood, and it treats the body as an energy system with a budget.
Yeah.
You eat food, you get calories, that's your income. You could spend that energy on only a few things. Basic metabolism, reproduction, and what he calls somatic maintenance.
Somatic maintenance?
Preparing your body, fixing damaged DNA, clearing out cellular waste like amyloid, fighting off infections, all the upkeep.
And it's a budget, so if you spend more on one thing, you have less for another.
Now, let's step out of the human world for a second. Think of a mouse in the wild.
Okay.
In the wild, 90% of mice are dead within a year. They get eaten by an owl, a cat, they freeze, they starve. It's a brutal existence.
So if you're nature designing a mouse, what's your priority?
Exactly. Do you invest a ton of precious energy building a mouse body with a flawless DNA repair system that could last for 3 years? Do you give it a robust system for clearing plaque from its brain?
It's a total waste of resources. The owl's going to get it in 6 months anyway.
You'd be building a Rolls Royce to take to a demolition derby.
Right.
So instead, you build the cheapest, most disposable body you can, a biological Pinto. And you pour every single spare calorie into one thing: rapid reproduction. You design that mouse to be able to have babies at 6 weeks old. The body, the soma, is disposable. The only thing that matters is the germline, the DNA you pass on.
So how does this apply to Sarah? We're not mice. We live for 80, 90 years.
We do now. But for 99% of human history, we lived in that same brutal world. We died from infections, from childbirth, from a broken leg, from predators. A lifespan of 40 or 50 was a good long life.
So our bodies were built with that expectation, a 40-year warranty.
A 40 or 50-year warranty, at best. So Sarah is operating a biological machine that was designed to be in a scrapyard 45 years ago. And the brain is the most expensive, high-maintenance part of that machine.
It's 2% of our body weight, but uses 20% of our energy.
So maintaining those neurons, keeping the connections clean, repairing their DNA, running the amyloid clearing systems, that is metabolically costly. It's a huge line item on the body's energy budget.
And after 50, the Evolutionary Budget Committee says, nope, we're not funding that anymore.
Why would it? In the ancestral environment where the budget was written, Sarah wouldn't exist. So the body just lets things go. It downregulates the production of protective proteins like cystatin C. These cellular cleaning systems become less efficient. The factory essentially stops paying for maintenance because the building was scheduled for demolition decades ago.
That Alzheimer's isn't a disease in the way we normally think of it. It's the default state of a machine running past its engineered lifespan.
It suggests that longevity itself, the very thing modern medicine has given us, is the single greatest risk factor. We've created a world where we can outlive our own blueprints.
Because it's not just that we're living longer. We are living in a completely alien environment compared to the one we evolved in.
The great mismatch.
And this is where the story of amyloid itself gets a crazy twist. We've been talking about it like it's just garbage, cellular exhaust.
What if it's a weapon? This is the antimicrobial protection hypothesis. And for me, it is one of the most compelling new ideas in the entire field. It changes everything.
So if amyloid isn't trash, what is it?
It's an antimicrobial peptide. It's part of our innate immune system, and it is incredibly good at killing bacteria, viruses, and fungi.
Really? The villain is actually a superhero?
Potentially. The gene sequence that produces amyloid beta have been conserved for something like 400 million years of evolution. You find it in most vertebrates.
And evolution does not keep useless garbage around for 400 million years.
Never. It would have been edited out long ago. A pathogen, let's say a herpes virus or some bacteria, manages to get across the blood-brain barrier.
An intruder.
An intruder. The brain's immune cells, the microglia, detect it, and they respond by secreting amyloid beta. The amyloid then acts like a sticky biological net. It swarms the virus, binds to it, and clumps all around it.
It encases the invader in a tomb.
It's a process called agglutination. It physically traps the pathogen so it can't infect your neurons. The plaque in this model is actually a sarcophagus for a dead or neutralized invader.
And then in a young, healthy brain.
In a healthy system, the rest of the immune crew comes in, sees this plaque pathogen bundle, and clears it all away. The plaque is temporary. It's a tactical response to an acute threat.
Okay, so where does it go wrong for us?
It goes wrong because of the mismatch. And this brings us to one of the most fascinating human populations on Earth, the Tsimane people of the Bolivian Amazon.
I read this study and my jaw was on the floor. The Tsimane are forager horticulturalists. They're living a lifestyle that's much closer to our ancestors. Lots of physical activity, no processed food.
And, critically, they live in a high pathogen environment. They have a high load of parasites, intestinal worms, chronic low-level infections. If you measure their blood for markers of inflammation, things like C-reactive protein,
they're off the charts. By Western medical standards, they should be chronically inflamed, which we're always told is a huge driver of Alzheimer's.
Exactly. And to make it even more confusing, they have a high prevalence of the APOE4 gene, the so-called Alzheimer's gene.
So they've got the bad gene and they've got the inflammation. Their brains should be riddled with dementia.
But they're not. Researchers took scanners into the field. They did cognitive tests. The Yosemite have the lowest rates of dementia of any population ever studied. Their rate of brain volume loss as they age is dramatically slower than in the US or Europe.
Possible.
It's possible because their immune system is busy doing the job it evolved to do. Their inflammation is hot. It's directed at a real physical enemy. Their amyloid beta is likely being deployed to trap microbes and parasites from the environment. It has a purpose.
Okay, now contrast that with us. We live in sanitized environments. We have clean water. We don't have worms.
But we have chronic, low-grade sterile inflammation. It's triggered not by a pathogen, but by our diet, high-sugar processed foods that lead to insulin resistance. It's triggered by a sedentary lifestyle. It's triggered by air pollution. These fine particulate matter particles get into our bloodstream and our brain.
So our immune system is constantly being poked, constantly being put on alert, but there's no actual invader to fight.
The brain senses all these inflammatory signals, these cytokines, and it thinks it's under attack.
Yeah.
So it does what it's supposed to do. It releases its weapon, amyloid beta.
But there's no virus to trap.
There's no virus to trap. There's no bacteria to encase. So the amyloid is released into the brain. And because our clearance systems are also often impaired by that same poor metabolic health, it just clumps to itself. It's friendly fire.
We're dropping bombs on our own headquarters because the alarm system is faulty.
That's the mismatch hypothesis in a nutshell. The Yosemite are using their weapon for its intended purpose. We are turning it on ourselves because our modern lifestyle is sending a constant false signal of invasion.
But I feel like there's one piece of the puzzle that we need to address. This idea of the disposable soma that we're meant to be scrapped at 50. It's powerful, but if that were totally true, why do we live so much longer than, say, chimpanzees? Why do grandparents even exist?
That is the essential complication. And it leads to a really beautiful refinement of the theory. If we were truly disposable right after our last child was born, human women would die in their 40s, like chimps do. But we have this long, distinct post-reproductive lifespan.
Which led to the grandmother hypothesis?
The grandmother hypothesis? Yep. The idea is that in our ancestral past, older women who lived past their childbearing years were not useless. They were incredibly valuable.
So?
They would help forage and gather food. They would provide childcare for their daughters' children. This support meant their daughters could have more babies closer together, and more of those babies would survive.
So a grandmother actively increases the reproductive success of her own lineage. She's still passing on her genes, just indirectly.
And that created a new selective pressure. Suddenly, evolution had a reason to keep the machine running past 50. It selected for women who could live to be 60, 70, and remain cognitively intact enough to be useful.
So we must have evolved some new tricks to protect our brains for longer than other apes.
We did. This is where we see what's called compensatory evolution. For instance, there's a gene that codes for a protein called C-glyc11. It's expressed on our microglia, the brain's immune cells.
Okay.
Humans have a version of this gene that is much more active than the one in chimpanzees. And C-glyc11 acts as a brake. It helps to dampen neuroinflammation. It tells the microglia, calm down, don't overreact.
So nature gave us an upgrade package, a sort of extended warranty for our brains so we could be useful grandparents.
It gave us a patch, a software update. But the problem we face now is that the overwhelming inflammatory signals from our modern mismatched environment, the pollution, the diet, the stress, are overpowering the patch. The inflammation is just too strong and too constant for C-glyc11 to handle on its own.
Our modern lifestyle is a bug that's crashing the new software.
We're caught in this evolutionary tug of war. The grandmother hypothesis pulled us towards a longer, healthier cognitive lifespan. But the modern mismatch is pulling us right back towards dysfunction.
So let's bring this all full circle. Back to the cancer patients and cystatin C. If this protein is the magic bullet, the elite cleaning crew that these patients have by accident, can't we just use it? Can't we bottle it and give it to people with Alzheimer's?
Our question, and it's the cutting edge of therapeutic development. On the surface, the logic is perfect. If a surplus of Cystatin in the blood protects against Alzheimer's, let's just make more of it and inject it.
But I'm sensing a very big but.
But remember the seesaw.
Right, cancer on one side, Alzheimer's on the other.
Cystatin C is often part of a larger biological program of cell survival, growth, and proliferation. If you take an elderly person and you systemically flood their body with high levels of these kinds of proteins, you might clear the amyloid from their brain.
And you might wake up a dormant tumor in their colon.
You risk tipping the seesaw violently in the other direction. You could, in theory, cure their dementia and give them metastatic cancer. That's the fundamental danger of messing with these deeply embedded pleiotropic pathways. You pull one lever here, and a trapdoor you didn't know existed opens up over there.
So a systemic treatment is too risky.
Precision. We have to be biological snipers, not carpet bombers. We don't want to dose the whole body. We want to find a way to activate these cleaning mechanisms only inside the brain.
How would you even do that?
Well, there's some really exciting work on a receptor called TREM2.
TREM2?
It stands for Triggering Receptor Expressed on Myeloid Cells 2. It's a receptor that sits on the surface of your microglia, the brain's janitor cells. Activating TREM2 seems to kick the microglia into a higher gear. It makes them better at clearing waste, including amyloid.
So you're not adding the cleaning crew, you're just waking up the crew that's already there.
You're giving them a strong cup of coffee and a new set of tools. The hope is that we can develop a drug that targets TREM2 specifically within the brain, telling the microglia to ramp up their use of things like Cystatin C locally without sending a grow signal to every other cell in the body.
You're trying to cheat the trade-off, get the benefit of the antagonistic gene without paying the price.
We're trying to decouple the two ends of the seesaw. It's incredibly difficult because evolution has wired them together so tightly. But understanding that Alzheimer's isn't just random chaotic decay, that it's the failure of specific conserved maintenance programs, gives us a much better roadmap.
It changes the entire goal. We spent, 40 years trying to shut down the factory that makes amyloid.
Right.
But cancer research and all this evolutionary theory suggests the factory isn't the real problem. The problem is that the cleaning crew got laid off.
Or that the cleaning crew was always scheduled to be laid off because the building was designed to be demolished anyway.
That's a profoundly sobering way to look at it.
But it's also empowering because it points us towards a new strategy, restoration, not just demolition.
So let's just try and pull all these threads together. It's a huge, complex story we've told. We started with this bizarre statistical anomaly.
That cancer seemed to protect against Alzheimer's. And that led us to the specific mechanism of Cystatin C, the protein that acts as a chaperone to prevent amyloid clumping.
And we zoomed out to ask why. We met Elias, the embodiment of antagonistic pleiotropy. The idea that the genes that make us strong and fertile when we're young, like P53, are the very same genes that cause breakdown and decay when we're old.
A direct trade-off for reproductive success. A deal written in our code.
Then we met Sarah, the face of the disposable soma theory. The cold economic truth that nature only invests in maintaining a body for as long as it's useful for reproduction. We are cars built with a limited warranty.
And finally, the environmental mismatch. The shocking idea that amyloid isn't just garbage, but an ancient weapon against microbes.
A weapon that's now misfiring constantly in our clean, modern world, triggered by diet and pollution, causing massive friendly fire damage to our own brains.
A system perfectly adapted for a world that no longer exists.
When you put it all together, it completely reframes how we should think about a disease like this. We see it as a failure, a flaw, something broken in the machine.
An enemy to be defeated.
But what this deep dive suggests is that it's not a flaw, it's a feature. It is the baked-in, calculated cost of the very systems that give us life and vitality in the first place.
You can't separate them. The light of vitality casts the shadow of decay. The cellular aggression that builds us up is the same aggression that, when its purpose is fulfilled, begins to tear us down.
We are not broken, we are simply finished. And perhaps the greatest irony of our existence is that the biological price of a mind that can understand its own mortality is a body designed to eventually force us to face it.
A profound paradox. The cost of a complex mind is a vulnerable brain.
Thank you for walking us through the biology of that paradox.
It was my pleasure.